Hello, class. So this is part two of the fatty acids and triacylglycerol metabolism series. So recall in the first part of the series, we just talked about the lipids that we ingest in our bodies, how it is eventually stores triacylglycerol in the tissues of our body to be used for later as a fuel source. So part two is talk about that process of how is this triacylglycerol actually the process of it becoming fuel for our body. And so the use of fatty acids as a fuel source requires three stages of processing. The first stage is the mobilization of the triacylglycerol, where it ends up being degraded into fatty acids and glycerol and ready to be mobilized into our bloodstream, where this transporting protein, serum albumin, transport the fatty acids to the tissues that is requiring energy. But in order for the tissues to receive these fatty acids, it needs to be activated and then transported into the mitochondria. And then from there, once it's successfully inside of the mitochondria, it goes to a degradation stage, where that's that third stage where it's broken down into acetyl coenzyme. And so this acetyl coenzyme is then processed in the citric acid cycle. So this is the process that is going to be described in this series, where you have the triacylglycerol that's in this fat droplet, where it eventually gets degraded, and then from there, once it's broken down into free fatty acids and glycerol, it can now successfully be transported into the bloodstream, and this is the serum albumin that attaches to the fatty acids for it to be transported in the blood. And then from there, in order to get into the mitochondria of the tissues that is requiring energy, it gets activated and then transported into the mitochondria, and then from there it goes through the beta oxidation and used as a energy source. So we're going to talk about this whole thing, but first do the first stage is when it gets initiated because of low blood glucose levels. So hormones such as glucagon and epinephrine is secreted in response to low blood glucose levels and binds its receptor in the adipocyte membrane, which is depicted right here, and then thus gets activated. Thus, when it does do that, it activates the adenylate cyclase, which will then convert ATP into cyclic AMP. And so once this cyclic AMP is created and there's an increased level of it, that stimulates the protein kinase A to start this phosphorylation type of functionalization on to the hormone sensitive lipase, where you see right here, this hormone sensitive lipase has undergoes phosphorylation, and you have this phosphoryl group that's on its surface. But not only does phosphorylation happens on the hormone sensitive lipase, but also on this fat droplet, the surface of it, which is also called the paralipin, you see you have phosphoryl groups that's on the surface of this fat droplet. But what's also on the surface of this fat droplet is another protein. And so as this phosphorylation is happening, now it's causing dissociation of this protein away from the surface of the paralipin. And then it goes and interacts with the ATGL, which is another enzyme that now becomes activated and now can start doing some lipase activity. So now that this ATGL is activated, now within this fat droplet, the triacylglycerol is attracted to the ATGL and then becomes degraded to produce a diacylglycerol and also a fat acid. But then the diacylglycerol is broken down even more due to the hormone sensitive lipase that has been uh, went through phosphorylation, and so now it's activated to now break down the diacylglycerol into a free acid and monoglycerol. And now we have monoglycerol. We go through one more stage of degradation where you have now the free glycerol and the free fatty acid that now can successfully exit out of the tissue as fatty acid and attach to a serum albumin protein that can now take this fatty acid into the bloodstream. But also what goes into the bloodstream is glycerol. And so this glycerol gets absorbed into the liver and gets phosphorylated. And that is happening right here, where you have this glycerol kinase, where it gets phosphorylated by ATP dependent type of enzymatic activity and form an L-glycerol 3 phosphate, and then from there goes to glycerol phosphate dehydrogenase to produce a dihydroxyacetone phosphate, and then goes through another enzyme activity of the triose phosphate isomerase, and then produce the D-glycerol aldehyde 3 phosphate, which should be very familiar because this was all part of the glycolysis that was shown last semester during biochemistry part 1. And so this is just a depiction of this mobilization of first that triacylglycerol is in the fat cell, broken down into fatty acid, and glycerol can now be transported into the bloodstream. And so this glycerol goes into the liver cell and then goes through glycolysis to produce pyruvate, or it can go through the gluconeogenesis and actually produce glucose. And as far as the fatty acids, that can be transported into other tissues where the fatty acids go through oxidation, where it gets broken down into acetyl coenzyme A and then goes through the citric acid cycle, thus producing carbon dioxide and water. So, in order for this fatty acid to actually be transported into the mitochondria, remember that the fatty acid needs to be activated and how it gets activated is it gets attached to a coenzyme A, and then from there you have this carnitine, which goes through an enzymatic activity where the fatty acid is attached to the carnitine, and then from there, once it gets transported into the mitochondria, and then from there, the fatty acids gets reattached to the coenzyme A, and then from there goes through the beta oxidation, and that carnitine that is now free gets transported back out to the cytosol area to be ready to start the process all over again. And so we'll break this part of how this fatty acid, first of all, gets activated by being attached to this coenzyme A. And so what happens is it goes through this enzymatic activity called the acyl coenzyme A synthetase. And so it goes through two-step process in order to finally get to this activated form of the fatty acid, and notate that this is an ATP dependent type of enzymatic activity, releasing AMP and a phosphorylated group. And so the first stage is this fatty acid actually gets bound onto ATP, where it is bound to the phosphoryl group of AMP, because it's AMP because two of the pyrophosphate group is released, and so now it's only remaining as one phosphoryl group, so that's why this is AMP, and that fatty acid is attached to the oxygen of the phosphoryl group. And then from there, the fatty acid then goes to the sulfur atom of the coenzyme group, thus releasing the AMP to be free, and now the fatty acid is attached to the coenzyme, which this is called the acyl coenzyme. So now this fatty acid is activated. And then now, what I mentioned before, the carnitine is used as a way of transporting the fatty acid into the mitochondria. Just notate that this carnitine is a zither ion type of compound, having both a positive and a negative charge within the same compound. So you have this hydroxyl group that is free and can interact and be functionalized, where the fatty acid gets attached to that hydroxyl group, and then now you have your free coenzyme A that's ready for more to ready to activate other fatty acids. But now we have this acyl carnitine that is now ready to start getting transported into the mitochondria. And so what happens is when this carnitine is attached to the acyl coenzyme, this is an enzymatic activity called carnitine acyltransferase 1. And you have this now fully activated fatty acid that can be transported. And so what initiates this, helps out with this transportation, is another protein called translocase. Now do you have this acylcarnitine, where the carnitine and the fatty acids are bound together, gets transported into the mitochondria, and now for part two, where the carnitine acyltransferase 2 now does the opposite, where it produces carnitine that is free now and binds the fatty acid back to a coenzyme A that is within the mitochondria. And now you have the acyl coenzyme A that's ready to be broken down into the um, beta oxidation of the fatty acid. But this free carnitine gets transported back outside um, of the mitochondria back into the cytosol and ready to start the process all over again of being attached to an activated fatty acid that's attached to a coenzyme A, and the process going to cycle over and over. And so this is the same depiction of what I spoke about before, where you have the fatty acid that's now in the cytosol. It gets activated by being attached to the coenzyme A, and then from there, uh, the fatty acids get transferred onto the carnitine, and then from there it can be transported into the mitochondria, and then from there be reattached to a coenzyme A again, and then goes through the beta oxidation for degradation and use of energy within the tissue. And so what we did thus far is the first stage, we talked about the fatty acid being being activated by being attached to the coenzyme A, going through two-step process where the fatty acid is first attached to the ATP, where it is technically AMP and fatty acid that is attached, and you have the pyrophosphate group that is released. And then while the fatty acid is attached to that AMP, it goes through a enzymatic activity again, and now being transferred onto the coenzyme A, and that's where you have that acyl coenzyme A, and now it's officially activated. And then once again, once it's activated, now it needs to be transported by being attached to the carnitine. So the acyl coenzyme which has the fatty acid attached to the coenzyme A. Now the fatty acid is attached to the carnitine, making acyl carnitine, and now can successfully be transported into the mitochondria, and then from there it can go through the once it's in the mitochondria, it gets reattached to a coenzyme A and then goes through the beta oxidation. So the beta oxidation part is part 3 of our series. So this is where we will stop. Where this the second part of the series is this talk about this: how do this fatty acids get mobilized and be free. It goes to the blood system and then gets activated and then transported into the mitochondria, and then eventually be broken down into energy for us. And so we'll talk about that in the third part of the series.